Hi, my name is Rebecca Donoghue and my third year research honours project was a glycobiology based one with Professor Jerry Turnbull, and we investigated the effects that ageing has on heparan sulfate and base 1 activity. Heparan sulfate, or HS, is a complex molecule that is composed of between 40 and 300 individual sugar molecules, all bound together in a linear chain. Chemical groups called sulfates can be added to HS by enzymes called HS6STs. The number of sulfate groups bound to HS specifies its structure, hence specifies the molecules that it can interact with within the body. Base 1 is an enzyme that leads to the formation of Alzheimer's disease, but in healthy individuals, the number of sulfate groups bound to HS allows it to specifically bind and prevent base 1 from causing Alzheimer's disease. So because ageing is the biggest risk factor of Alzheimer's disease, we wanted to investigate whether ageing had an effect on the structure and the number of sulfate groups bound to HS, and whether this in turn affects its ability to bind and reduce the activity of base 1. We first investigated whether the number of HS6STs changed with increasing age. We did this by extracting DNA from 3-month-old and from 18-month-old mice, and compared the number of HS6ST genes present in each age group. We then investigated the structural changes of HS with increasing age. We did this by extracting HS from 3-month-old and from 18-month-old mice, digested it into smaller length chains, and then separated the shorter chains based on how many sulfate groups were bound. We could then compare the structure of HS between the two age groups. Finally, we added the extracted HS to base 1 and measured its activity. We then compared the activity of base 1 when bound to HS from the two different age groups. Firstly, we found that with increasing age, the number of HS6STs decreased. So in theory, we would assume that the number of sulfate groups bound to HS would decrease. However, this was not the case, as we found that the number of sulfates, hence the structure of HS, did not change with increasing age. So this suggests that other enzymes within the body work to maintain the structure and the number of sulfate groups bound to HS, independently of the number of HS6STs. We then found, as expected, that because the structure and the number of sulfate groups bound to HS didn't change, then its ability to bind and reduce the activity of base 1 also didn't change between the two age groups. And this is interesting because we know in Alzheimer's disease, the activity of base 1 does increase. So this suggests that the way HS is made in healthy aged individuals compared to the way it's made in Alzheimer's disease patients must differ in some way. And although this doesn't seem to be due to the number of HS6STs, there may be other enzymes within the body that are responsible for this difference.